haploid, it ends diploid. That information allows you to just plug in these numbers. So we know that it starts with 84, and at the end, because I know it's ending diploid, I still know that 84 is the number of chromosomes. So we can start with that. Now we have to think about the second box is chromatids. That's what that means, total number of DNA molecules. So we have at the beginning of mitosis duplicated chromosomes. So how many chromatids should be in that second box? 168. Does everybody see why that is? I like to sometimes draw this out like this so that I can keep track of, all right, I have sisters, so I have 84 and this is 168. At the end, what goes to each of the cells? The sisters come apart, right? So now I draw it like this. I have a single strand that goes into each cell. So how many chromatids should be there? 84. So this is 84. Okay, that one's pretty straightforward. The diploid to diploid, that kind of keeps it simple that, you know, the number of chromosomes is not changing. I'm just changing whether there's 2 copies or 1 copy. All right, so meiosis 1. How does meiosis 1 start? Anybody feel confident? 84. 84 chromosomes. It starts out diploid. And then how many chromatids are there in the beginning? Do they have sisters?
Yeah.
They do, right? And I will draw out just to show that at this point in time they're pairing up. So I have all of these chromosomes together, but there are sisters, so that's how I get 168. At the end, I go from diploid to, what's the ploidy number for? Should be 1N. So that tells me, this number tells me what to write in this box. What's the haploid number? 42. And what happened was these tetrads split. So now I still have sisters. That tells me when I see my X, I know, all right, I need to double that number to figure out how many chromatids are there. So what's the chromatid number? 84, okay? So that's the end of meiosis 1. This is going to be the beginning of meiosis 2. So I copy all this information over here, 42, 84, haploid, and I'm starting with sisters. At the end of meiosis 1, what is the ploidy? Is it 1N or 2N? Or something else?
You said at the end of meiosis 1?
Yeah, or meiosis 2.
Oh, 1N.
Still haploid. It's never going to go below haploid. This is one place a lot of students I see, like, I don't know if it's because they think about there's 4 cells, or sometimes I see different numbers at the end, but the ploidy, once it gets to haploid, it stays haploid. So we go from 42 with 84 copies. This number will not change because that should always match up with my 1N. So then what will go here in the chromatid number? Yeah, what's happening is the sisters are separating, so I still have 1 strand for every one of those chromosomes. So it goes 42 to 42. So another thing you could practice, so in addition to these, I'm just giving you ideas of what should I think about. So change the numbers, that's one thing you can change. Another thing you can do is think about other stages. So if we were looking at S phase, what would this look like? Or if we were looking at G1, what would that look like? So that's a good way, like any stage of the cell cycle, you can think about how would the number of chromosomes and chromatids change. All right, anybody have questions on this top box? Or if something doesn't make sense, if you have the question, I'm sure somebody else also has the question. Anybody want to clarify? All right, let's do the very bottom box then. Okay, so cell division. Does cell division happen for all of those?
Yes.
Yeah, it does. So we could say, put this in. So yes, we could say yes. This one we might say like there's a reduction division. That helps keep track of what is actually gonna happen. This one is an equational division. But really, you could say yes for all of those, okay? Chromosome reduction. So does that happen in mitosis?
No.
No. 2n to 2n, there's no change in that number. How about meiosis 1?
Yes.
Then we say yes, and just so that you remind yourself, you can put 2n goes to 1n. How about meiosis 2?
Oh, yeah. No.
No. It's 1n and it stays 1n. The only thing that changes is chromatid number, but that's technically not changing the number of chromosomes. Now this one, I'd like to hear what you thought for the genetic variation produced. In mitosis, is there genetic variation? No, hopefully not, because we're trying to make a clone. So we say no. How about meiosis 1?
Yes.
Yes, there is. And what processes generate the variation? Crossover. Can anybody think of another one? Yeah, so we didn't talk about it in our class, officially, but when the maternal paternal chromosomes line up, they can line up with maternal all on one side, or they could have different combinations, because that happens randomly. Mom or dad chromosome they could randomly align. So for every single chromosome pair that you have, there's 2 possible alignments. For humans, that gives us 2 to the 23rd power of different ways that these could align. So that's like a huge number of possible gametes that you can create. So there's 2 things here. 1, crossover. 2, random alignment. And what stage does that random alignment happen? In metaphase. So this is prophase. This is metaphase. 1, right, for both. Okay, in meiosis 2, what did you think? Is there genetic variation produced? Because I could accept either answer if you justify your answer. What did people think?
I feel like yes, because we're looking at I'm looking at the very beginning of meiosis, and so that's genetic variation. I feel like it's all
Was
part of the
produced.
process,
Mhm.
but then also.
Wait, so the events, yeah, go ahead, Matt.
Well, with the crossing over in meiosis 1, those get the chromatids get split, so that.
Yeah.
Goes from-
So that would be the reason why you could say yes. I could interpret this to say, was the process that generated genetic variation, did it take place during this phase? And the process that generated it did not take place during meiosis 2, but as Nat said, when we have these 2 strands, now erase this one because that's already separated, but when these line up, they could line up like this, or they could line up the opposite way. And if you have all of the crossover events like that, you could generate different gametes. So I think you can say yes. If you understand that part, or you could say, no, the processes that happened, they've already happened in meiosis 1. Okay, so for crossing over, where does that happen? The event itself,
Meiosis 1.
only in meiosis 1, we say no here, no here. And then this one, random distribution of maternal and paternal, because it actually says maternal and paternal chromosomes, there's only one phase where that happens. So which phase is that? Yeah, in metaphase 1. That's the only time the homologs are still together. So we should say no and yes and no here, because what we were just talking about is different than maternal paternal chromosomes. And then what lines up? So that's either asking, do you have individual chromosomes, or do you have the homologous pairs? So for mitosis, what lines up? Yeah, so I'll write individual chromosomes. But you're right, what's gonna split is the sister chromatids. And then in meiosis, then we would say it's the homologous pairs, or you could say tetrads. And then for meiosis 2, what would you say? Yeah, so you could say individual chromosomes, and then what's gonna split? Chromosomes, okay? what separates here is the sister chromatids. Here, it's the pairs, right, or the tetrads. And then, again, sister chromatids. Okay, so I think that's good for now. What you'll have to do on the back is, I'll let you do this because you can take your time. I don't want you to, don't go and look at a picture from a book. A lot of times people just try to copy what they see on the internet, and the thing that I'm asking you here is to draw a very specific thing. A diploid cell, that means there's maternal copy and paternal, so use 2 colors for that. You can decide, blue and red or whatever. And then 3 different chromosomes. So I would do size differentiation so that you can see, like, I have 1 big, 1 medium, 1 small in each of those sets. And so draw your own pictures, don't copy pictures that you find somewhere else. Try to go through this and show the events, like show crossing over. And you don't have to show all 4 cells. So once the cell splits off, like in prophase 2, you just have to show me one of the cells, not both of the cells. Okay, does that make sense for that part? All right, so we can take a little break. When we come back from the break, we can get started putting our L4s or whatever we have, I guess, if we have like adults, onto